Hello guys, good evening. Today we are going to discuss about necrosis. So, what is necrosis? Necrosis is a pathological process corresponding to the severe cell injury or tissue injury. Necrosis, which is characterized by denaturation of the proteins, and the cellular contents will be leaking to the extracellular space, leading to the local inflammation and enzymatic digestion of lethally injured cell. So let's discuss about it in detail. In these inflammatory cells, they will produce more number of proteolytic enzymes. Proteolytic enzymes, they will digest the proteins. So in combination of the phagocytosis and enzymatic digestion, usually leads to cleavage of the necrotic cells. Necrosis, which is associated with the leakage of intracellular proteins from the damaged cell membranes. Okay. Suppose in example, if there is any damage to the myocardium, then what will happen? Cardiac troponin levels will be increased in the plasma after 2 hours of myocardial injury. So how the morphology, how we can observe this necrotic cells means increased eosinophilia means uh, the cytosol of the cell will be deep pink in color because this RNA is not there. If RNA is there, there is no loss of cytosolic RNA. RNA is usually blue in color, like hematoxylin it will stain. So it is binding to eosinophil, but there is no RNA, so eosinophilia will be increased and accumulation of the cytosolic proteins will get accumulated. As a result of loss of glycogen particle will be getting lost, and glassy appearance will be observed as there is no fat. So and cytosol become vacuolated it will be appearing, and appears as some moth had eaten the cell, like that. Dead cells can be replaced by oral phospholipid aggregates known as myelin figures. What are myelin figures means these plasma membrane will be, no, plasma membrane consists of phospholipids. Phospholipids means like sphingomyelin, these are sphingolipids. So sphingolipids and phospholipids. Some myelins will be, no, these myelin figures will get accumulated in place of the damaged or dead cells. So these are phagocytosed by other cells, they degraded into the fatty acids. Fatty acids degraded into phagocytes. Deposition of the calcium rich precipitates. Marked dilatation of the mitochondria. Mitochondria will be markedly dilated. Okay, marked dilatation of mitochondria and appearance of large amorphous densities and aggregates of fluffy material representing the denatured proteins. So as we had already saw that denatured proteins will get accumulated. These will appear as amorphous densities, and DNA breakdown, nuclear changes will be observed. Basophilia means deep blue color of the chromatin. This chromatin deep blue color will be fading because of the karyolysis. Nucleus is getting lysed. Pycnosis means nuclear is shrinking, and basophilia, which is increased. Chromatin condensation will occur. And karyorexis, nucleus will undergo fragmentation with the passage of the time, half days, 1 to 2 days. The nucleus in the necrotic cell totally disappears. So, what are the different types of necrosis? As we saw, what are the changes in occurring in the necrosis? Like it is a reversible cell injury. We have to write these points. Like it is reversible cell injury, and these proteolytic enzymes will be getting out of the cell to uh, make the inflamed cell to die, like that we have to write. And in necrosis we see pycnosis, karyorexis, and cells will be swollen, moth eaten type. In microscopy we can see cytosol as eosinophilic, as there is loss of RNA. Okay, let's discuss further. It is different from apoptosis. Apoptosis we will see shrinkage of the cell, whereas in necrosis there is a swelling of the cell. In necrosis, what are the types of necrosis? They are coagulative necrosis, liquefactive necrosis, gangrenous necrosis, caseous necrosis, fatty necrosis, fibrinoid necrosis. What is coagulative necrosis? Coagulative necrosis, which is most commonly seen in many all the tissues except the brain. In brain, we will see liquefactive necrosis. First, we will discuss what is coagulative necrosis. This dead tissue which is not immediately phagocytized. So in these affected tissues will have film texture. So this injury denatures not only structural proteins but also enzymatic proteins. It blocks the proteolysis of the dead cells. Proteolysis won't be occurring. So these dead cells, they will get accumulated. So intensely eosinophilic cells with indistinct and reddish nuclei gets accumulated. So it will give reddish appearance. Necrotic cells which are broken down by the lysosomal enzymes. Lysosomal enzymes will break the necrotic cells which are derived from the infiltrating leukocytes. From leukocytes, these lysosomal enzymes which are derived, these necrotic cells which are broken down by them, and they will remove the debris of the dead cells by the phagocytosis. So if this debris remains, it will obstruct the vessel and leading to the coagulative necrosis. Localized area of the coagulative necrosis is known as infarct. We call the small localized area as infarct. So digestion of dead cells resulting in transformation of the tissue into viscous liquid. It is in contrast to coagulative necrosis. Dead cells will be digested and they will be transformed into liquid. So we are calling it as liquefactive necrosis. So it is usually seen in the brain. And it is usually seen as creamy yellow appearance due to the leukocyte infiltration. We usually call this yellowish discoloration as pus. Hypoxic death of the cells in the CNS will manifest as liquefactive necrosis. What is gangrenous necrosis? Gangrenous necrosis, usually limb gets affected. Normally gangrene will be three types. One is gas gangrene, wet gangrene, and dry gangrene. Wet gangrene and dry gangrene, which are commonly seen in diabetes patients. Blood supply to the limb gets occluded. So it is typically like coagulative necrosis involves multiple tissues. And bacterial infection which is superimposed, more liquefactive necrosis will occur. Okay. Next we will go for caseous necrosis. Caseous necrosis we will see in the tuberculosis. Appearance friable white. On microscopic examination, necrotic area appears as structureless due to the fragmented or lysate cells. Center we will be seeing epithelioid cells, and surrounding we will see Langhans cells or giant cells. These epithelioid cells will fuse to form the giant cells, and this is like as we see in gross, we will see white color cavities in the lungs, and this is known as granuloma formation, which is seen. Fat necrosis we usually see in pancreatic tissue due to the pancreatic lipases stimulation due to fat destruction. These pancreatic lipases enter into the peritoneal cavity, and we will observe them. It has small white color appearance. Okay. Associated with the acute pancreatitis. These acinar cells will be present on the pancreas, which are secreting the enzymes. As we saw in pancreas, both endocrine portion and exocrine portion. In exocrine portion, these acinar cells which are present. These damaged acinar cells will be leading to the leakage of the pancreatic enzymes. These liquefy the membranes of the fat cells in the peritoneum. And release of the triglyceridases splits by the pancreatic lipases. Fatty acids and along with the calcium, they will be appearing as chalky white appearance. Necrotic areas, necrotic fat cells, basophilic calcium deposits, and inflammatory reactions. Fibrinoid necrosis, we will see immune complex deposition in the blood vessels. This is observed in even in SLE, as it is Arthur's reaction. Because of immune complex of the antigens and antibodies in the walls of arteries. Immune complexes, plasma proteins, they will be leaked out of the vessels, bright pink and amorphous appearance, will be known as fibrinoid.